Now we're going to shift from developments in cancer care to more the personalized precision approach that we're seeing more often these days. And here comes Marty. Come on up, Marty. You're just in time.
And thank you, Roger, and welcome to the panel. I wish someone had told me I can't use Apple Pay with an American Express card on the parking app, or I would have been on time. So I'm a computer scientist and an entrepreneur and a visionary, and was lucky enough to be in Silicon Valley during this amazing golden age, which continues to this day. And robots I built in the 1970s are in the Smithsonian and the Computer History Museum. I started the first company that was focused specifically on electronic commerce. We hired Mark Andreessen and brought him to the Valley. He spun Netscape out of the company, which was called Enterprise Integration Technologies. And we started the association, the alliance, that made e-commerce possible by taking all the small companies, and Netscape at the time had like 20 people at it. Amazon had even less. We brought them together so they could work with each other. And so we could bring in the FedEx, the banking, the shipping companies, and so forth, in order to be able to make all of commerce happen end to end. And that's kind of the situation we're in today with precision oncology. We have a lot of the pieces, they need to be connected. In 1998, I was diagnosed with melanoma metastatic to liver and given 9 months to live. That was before any of the targeted therapies and checkpoint inhibitors, which were developed in melanoma simply because there was nothing else in that disease. It was really deadly. My life was saved by a clinical trial that failed. That's where luck came into it. But when I survived, I kind of pivoted to a much more significant undertaking, which was trying to use my skills as a computer scientist and an entrepreneur to make a difference. And I started a company called Collabrix in 2008, which was the first company to offer precision oncology navigation as a service. And we were using RNA-seq in those days, using Effie chips. 3 years later, I spun out Cancer Commons, which is a nonprofit that helps patients who have exhausted the standard of care navigate the cancer maze. And over the years, we've helped over 10,000 patients. And I'm very, very proud of that. But it's a drop in the bucket compared to what's needed. The need is kind of 10,000 patients a month. And that was beyond my managerial skills. So I'm very pleased to say that as of last Monday, I gave up being the CEO of Cancer Commons, and I brought in a really experienced leader who will be here this afternoon, and I hope you'll all take the time to meet him. That's Cliff Reed, who, among other things, did Complete Genomics, took it public, and more recently Travera, which is a functional testing company. I'm going to run Cancer Commons Labs, which is kind of Marty's sandbox, to be able to work with all of you. Cliff has a very ambitious goal. He's aiming for 100,000 patients a year, and he aims to get there by using AI to curate all of the advanced treatment options and match patients to them, and then track the patients to see what works. It's something I aspired to do, and it was beyond my pay grade, but Cliff's going to do it. But we can't do it without your help. And the reason I say that is because our goal is not just to curate the options that are out there, but to create new options. And that's where all you guys who are investors and startups come in. And I want to work with you all in labs in order to be able to engage you, bring you to clinic much faster than you're able to do. And I'm sorry. Okay. Could everyone hear me now? All right. And be able to accelerate your progress, give you the data that you need to be able to train and validate your algorithms, for example. But most importantly, bring these things to the patients much sooner. So at this point, I would like to introduce the panelists and simply say that precision oncology, personalized medicine, is really something that's needed on multiple levels. Obviously, the patients need it, but so do the people developing drugs, because as you all know too well, developing a new cancer therapy, or a new test for that matter, is extremely expensive. It's time consuming. It's risky. You know, the number I hear, and I don't understand how this number happens, is an average of 2.6 is the latest number, $1 billion to bring a new cancer drug to market over more than a decade. And the trials where most of this money is spent are focused on population statistics. So whether the drug will do any good for a given patient is a crapshoot. So I'm going to turn it over to you guys now, and we'll have a discussion.
Hello to everybody. My name is Brad Niles. I'm CEO of Arise Precision Medicine. We're a Davis-based startup working in the siRNA field. And so where we got our start is having family that had gone through their cancer journey, seeing the side effects that come from a lot of these toxic therapies, and really a goal to create something better in which the patients could eliminate their cancer without killing off all their normal healthy cells and all the side effects that come along with all of that. And so really to get to a side effect free type of cancer therapy, we needed to go directly at the source of the cancer. And we were fortunate at the time to identify some really exciting research that was coming out of San Diego with an individual that had identified one of the early changes in cancerization of the cell, epigenetic regulators. So long before any of these downstream mutations occur, you have changes in gene expression that drive a lot of these pro-signaling pathways. And so we picked up where this investigator had left off, developed a therapeutic approach. We identified siRNAs as the best modality to go after this particular target in order to get some uniqueness in between a truncated version of the gene that was in the cancer cell versus the normal healthy tumor suppressor version. And so we spent the last couple of years working on a drug development project. We're in the preclinical stage. We've taken our siRNAs from in vitro, paired it with a targeted drug delivery system. It's a calcium phosphate nanoparticle. Allows us to get specifically down to the side of the tumor, get better uptake into the cell, and allow these siRNAs to do their mode of action. So.
What's the time scale? When's that come in the clinic?
So we hope to get our IND application done in about the next 2 years and start shortly thereafter.
How long have you been working on it?
We've been working on it about 5 years at this point.
2.5 billion.
Hi, everyone. I'm Laura Shea, the founder and CEO of Tipping Point Biosciences. We're a UCSF spinout out of Dr. Geeta Narlikar's lab at UCSF, who's my co-founder. And so we're also an epigenetic therapeutic company. And so one of the biggest problems in targeting epigenetics is that most people have targeted single epigenetic factors, but these factors have roles both in normal cells and in disease cells. And so in order to get better specificity, we came up with a different idea and technology to really target these epigenetics and reprogram the epigenome in disease in a very specific way. So we developed a platform technology that recapitulates the epigenetic proteins actually in the context of how they function on chromatin. And so the key is that a lot of the folding and the interfaces that are now accessible when they're in the right context in the on the genome is actually very different in disease versus healthy cells. So we're trying to drug these unique interfaces. So this is something that, AlphaFold can't model yet because we don't have the data. So if we can generate these networks of interactions to understand the new types of interfaces that are unique in disease, we think we can get better therapeutics and more personalized medicine for specific networks that and specific epigenetic networks that go awry in disease and in cancer.
This is personalized medicine on a long time scale, isn't it?
Yes.
We're going to get back to more specifics in a second.
I am Amy DeGolier. I am the founder and CEO of Represent. And we are building the missing human signal that's been lacking in precision medicine. Essentially, precision medicine has made incredible advances, but when you're using the genomics, you're using the imaging, you're using the labs, you're using what's in the EHR to train out these AI models, there's a really crucial piece that we've overlooked, which is the 99% of the time that patients are apart from the clinic. And it's how the patient is experiencing their treatment. It's how the therapy is working within the patient itself. And the challenge is you may, put together the best treatment for this individual patient, and they go home and they go septic because for that patient's individual biology or something that happened home, it all went awry. What we're doing is capturing this lived experience in a dense, frequent, structured way. We're applying ML to find the patterns within it, and over time using these signals to identify things like progression of disease, efficacy of treatment, and patient risk at large. In the first order, we've created a system that really helps patients personalize their own care so that they use our system deeply so that, they get better control of their own symptoms. Their doctor can better adapt their care. But in the long run, we're looking really to develop this whole layer of precision medicine to make the entire field more accurate.
And that's an example of just one of the many pieces of missing infrastructure that's needed in order to be able to bring precision medicine to market. So I think at this point, let's try to dive deeper into specifics. And I'm looking here for challenges that you faced in your companies and ways to overcome them, specifically around things like funding, regulatory processes. How can we make the whole clinical development process, clinical trials, much more efficient? And what other pieces of infrastructure are missing? For example, Cliff Reed, who had run a functional testing company, was stymied by the inability to be able to get specimens from community hospitals, or even from Stanford for that matter, within 24 hours, so they were still fresh and he could do his testing on them. There's a lot to be done. So you guys, aside from whatever you're doing at the moment, are all very experienced. And make us smarter. Whoever.
Yeah. Well, I'll dive in on the clinical trial piece just for a second, because that's actually where we've been leaning in very heavily recently. There's so many more toxic therapies coming to market, and it's wonderful to bring these therapies that are needed. But the challenge, if you're a drug developer, is, you need to move fast and you need to move fast safely. And if you get a high-grade toxicity as part of your trial, the whole thing could be over, and this incredible treatment that's needed for so many patients could be lost. And so as an organization, to really develop more signal and to drive volume, that's where we've leaned in to help trial developers, identify these AEs sooner, to lower these high-grade toxicities, to make the treatment more tolerable. Because if we aren't able to get these drugs out to the patients that they might be relevant for, we're really stalling development in that long-term development piece that you mentioned. Overall, we're not making the progress we need. So in the first order, I think that's the first application that we're using for this precise and personalized element. But over time, it's then, of course, to identify which patients it may be suited for in the first place.
Anyone else want to comment on trials?
Well, I mean, kind of related is I think that as a drug developer, I think that it's really important to understand why drugs fail in the clinic. And unfortunately, a lot of the clinical trial data is not accessible, right? And so I think that makes it more challenging. If we could be more open source and share some of this data, it would be a lot more helpful in terms of understanding some of the barriers that we need to get over early in development, before making similar mistakes when we actually run the clinical trials.
Yeah, wholeheartedly agree with that, and wish there was a better system. Maybe somebody can figure that out. But when we started our drug development process, we tried to work backwards from clinical trials with the idea, we know what we want it to look like when it gets to the clinic. How do we reverse engineer that down to the most basic levels? And the primary function or the primary point to consider there was failure due to off-target toxicities. And so an sRNA approach we believed could avoid a lot of those potential pitfalls with off-target. And sRNAs and RNAs in general have been kind of maligned over the years. One of the early issues with sRNAs was an off-target in terms of the complementary sequence. Where sRNAs have really advanced since their discoveries was in the delivery. And so our major focus was not only to design the most effective and most on-target sRNA, but actually be able to get it specifically to the site of the cancer. And if you're not getting it taken up into normal healthy cells, you're not going to have those side effects. And so our major focus was in nanoparticles, not only just like the LNPs that a lot of us are familiar with that often go to the liver. How can we get beyond the liver? How can we talk specifically to this? Interesting things going. Still on. How we can get it specifically to the site of the tumor. Is the oh, maybe it's a battery. Lung cancer is our cancer of interest. Because of a huge market in those patients that have the defect that our drug goes after. So if we can select patients that will respond to the treatment, if we can get the treatment in high enough levels down to the site of the tumor and only get uptake into those tumor cells, we can avoid a lot of the failures that others have dealt with.
All right, since you challenged in the first thing, anyone has a better idea, I want to throw out a better idea.
Excellent.
It's very provocative. It's a complete paradigm shift. It's flipping the goal of trials from approving drugs to curing patients. And I'm thinking that this could address a lot of the issues that we're facing, but with a very big caveat, which is that this is really not aimed at first-in-human drugs, because those should be done in a phase 1 unit for sure. But if you think about starting with the patient, right? And we at Cancer Commons, all of our patients have exhausted. They don't come to us till they've exhausted all the standard care, right? And then we have to find something for them. And the idea of starting with one patient and really doing precision oncology on that patient, working them up, doing all the omics, all the functional testing that you can, doing computational and systems biology, even digital twin, to try to do the best job that you can. Every patient is going to be different with advanced cancer, right? There is no trial. You can't, right? It's all about trying to find what the optimal combination of therapies is. And it almost certainly, I make a very provocative thing. No one knows how to use the thousands of drugs that are available in an optimal way for a given patient, right? That's precision medicine. And so if you do something and it works, what you're really perfecting as much as whatever combination of drugs is the technologies that you're using in the core of all the people out there who are developing tests and services to help eliminate the guesswork, right? So you get this patient and if the patient doesn't respond, you get another shot on goal. You try something else for that patient, right? But you're testing combinations of things, which is not normally done because there's too many of them in a trial, and they want to get a pure signal on something. So you're curing the patient. There's no inclusion, exclusion, no randomization or travel required. Everything is focused on this patient, right? And then for you investors, you get to see if it works on one patient. That doesn't tell you anything, but it tells you one thing very important, which is that there's a there. And the scientific risk suddenly goes much lower. And also the market risk goes lower because this is something that the top physicians that we work with wanted to use for this patient. Eliminate market risk, eliminate scientific risk, and be able to then buy another vowel, so to speak. Invest a little bit of money to try it on a handful of more patients, whatever it is, the technique and the therapies, right? So you've made a very small bet, and you can make a large number of small bets for a given set of bucks. I think this is a better way to do clinical development and investment by flipping the paradigm. And I would really appreciate constructive feedback or even destructive feedback on this radical idea, because I'm intent on doing it and doing it commercially with many of you. And the audience can chime in here if someone has something to say.
Not a criticism, but more of a question. How do you manage the side effects that come from each one of those drugs when you're, technically stacking side effects?
I'll take that one, Brad.
There she goes.
We just don't happen to have an expert on that in the panel.
We just do. And I would say, to expand, where this idea is going for you, Marty, I think the other element to keep in mind here is the access, how we don't just do this in a small area. This is precision medicine, but it doesn't have to be for a precise population. And I think that's how our complement is really helpful. If we can start to create almost a symptomomics, if we want to put an omics on that, and we can start to use these signals so that we can push this out to the edges of society, it becomes not just let's find the signal in the center, but let's push this out as far as we can. And I think, Brad, your point is, how do you do that with the symptoms? You need to manage it in a different
There's
way.
even an opportunity to do science in real time, right? All these scientists who do cancer research, and many of whom have never seen a patient in their lifetime, right? Get them to weigh in on the initial treatment, and when something is not working, either side effects or the effectiveness, right? To get another shot on goal, a knowledge turn, I guess. The patient's getting another shot on goal. And you can do 3 or 4 of those a year, as opposed to locking someone into a trial protocol for multiple years. Let's do something different. This is Silicon Valley. I'm ready. And I don't have a lot of time to do it either. Probably in the next decade. It's got to get done, guys. All right. So then the next question, we're talking about incremental ways of financing, place lots of small bets, double down on the winners. Any other ideas? Financing, all these companies that I work with that are developing therapies are really struggling at the moment because the funds are drying up. And what can we do to help those companies? Anyone in the audience? In back? Not a problem. All right.
That's pausing. A lot of people don't realize the opportunity in crowdfinancing and crowdfunding, and this seems like an ideal place to do it, because because I like to say crowdfunding is good for affinity investors. When you've got a product that consumers understand and they see the problem and the potential, I mean, this is a place that fits that, given that almost everybody knows somebody with cancer or has been affected by it. So that's one option. Marty, what is our panel? Someone in the back here. Malika, go ahead.
Someone earlier today asked me about free round and how you could support a company that someone else who's currently in the audience, they asked a little bit more about bridge rounds. So as you mentioned, as few companies are going through a tough funding environment at this time. And because they've already raised money from VC funds, they've gone through technical due diligence, they have strong leadership on their boards. They might have raised money from Bill and Melinda Gates Foundation, or I believe someone said that they're an ARC spin out. So yes, when you invest in a bridge round, you're coming in at a smaller check size than Bill and Melinda Gates are, but you are benefiting from the technical due diligence that they put the company through, or other big name investors. So just to give another example, I wouldn't totally call that crowdfunding, but I would still call it coming in with your own funds at a check size that's more realistic and doing it in a way that supports your own finances, because the company has gone through technical due diligence as well as major commercial or social support.
Yes,
You get a micro VC, so you're right here.
What do you like the bar? Everybody like the bar. See, yes, bar.
So are we hearing it?
all of the lung companies in Silicon Valley for over 20 years. And since I'm a collector of data, what I'm thinking about in a group like this is I might put a group within Link SV of just getting you all together connected to share ideas of what you've all learned, what you can contribute to each other, on who you might know who are investors, great advisors, great board members. I love all this stuff. My grandfather was the head of obstetrics at UC Berkeley, UC San Francisco in the 1900s. My dad was a doctor who basically was responsible for the labeling of prescription drugs signed by Ronald Reagan. My whole life is dedicated at this point to wanting to help advance things in medicine. But I'm an angel investor with a small amount of money. But I love the fact you guys are all doing things to make a difference. I can help you by maybe figuring out a way to get you to all share it together. I'm an expert at network.
Well, let me add one thing. There is a radical idea here too, which is to tap a different source of capital, right? People who are affected by cancer. And there are, whether you do it through crowdfunding or you do it through ultra high net worth family offices, right? People who have donor advised funds or want to invest their capital. I'm going to be setting up a cancer impact fund within Cancer Commons to give people the option, if they don't feel like investing, like donating philanthropically, which has fueled all of our work to this point, to be able to provide dry powder. So when we see something that's working in the clinic, again, de-risked both scientifically and market-wise, to be able to place, to have dry powder to rapidly double down on these winners. It's a much better way to do it than I think the way the industry has traditionally done it. And I would love to collaborate with the investors out there to set this impact fund up. A lot of capital, trillions of dollars that could be capped. Yes?
So I'd like to double down on your idea about talking to real patients because I like to share an experience. And maybe others could learn from that too. So we are a nonprofit trying to help cancer patients, really more oncologists. We have an AI-based tool which helps oncologists to give them standard of care. A close friend of mine for the last 2 years, 3 years, got diagnosed with stage 4 pancreatic cancer. And he's been at UCSF. He goes back and forth. Just 2 weeks ago, he's kind of now given up all chemo. So he came and, he used our system, and the kind of feedback and the emotions that we got, when he used it, he said, I wish I had it, when I was getting my chemo, because there's so many questions that I can't ask the folks, in the nurses. And a lot of time I go for chemo and they said, I'm not ready for it. I come home. So my point is that this notion that you're saying you should talk directly to patients, at least we could, because we're not kind of giving them any drugs. It was more of a information tool. But I think that really hits home when what you say, because that interaction completely changed the things for us from day and night.
And I want to work with you. I want to work with all of you. I got the 1 minute flash. So anyone else have something that they're desperate to say, please?
Well, I just want to share that I think this is a great idea to share experiences. We did a crowdfunding project while we were in the Cancer Hacker Lab. It's non-trivial. It's incredibly time-consuming. And I'd be happy to share all that experience. I think I can do it better next time. I've raised 6.5 million in grants. I didn't get the first one. So you're willing to learn. And the last thing I want to say is that I have been blessed with amazing investors that have supported us. And those angel investors are committed because of our mission and because we continue to update them. And by the way, you're talking about intelligent money and all of that jazz. They're pretty smart. They've done businesses. They have operational experiences. I talk to VCs with 0 operational experience. They are money allocators. They never worked in a company. So.
And 90% of the drugs that enter trials fail to become products. So,
Hey, Roger, I'll just Roger.
Okay.
Last comment.
Yeah, I'll just mention, for example, by using a keyword search in our system, if you typed in cancer, we'd come up with like 137 companies in the Bay Area with 20 years have been involved in cancer, and I have all the investors. So the point is I can figure out that and share with you who might be investing in something you care about by a keyword. That's it. That in itself is probably valuable.
Okay, and with that, I want to thank our panel. Personalized care.